Dear student, today we will talk about cellular respiration, in which we'll be describing glycolysis. Now, cellular respiration is the biochemical process in which there is oxidative breakdown, that is in presence of oxygen, oxidative breakdown of food, which is also known as respiratory substrates such as glucose, carbohydrate, protein, lipids, with the help of enzymes, the enzymes of cytosol. With the help of enzyme, with the release of energy, water and carbon dioxide. There are two types of respiration, aerobic and anaerobic. Aerobic, which occurs in presence of sufficient oxygen, where the food is completely oxidized, releasing energy, water and carbon dioxide. Anaerobic, which occurs in presence of insufficient supply of oxygen, the food is incompletely oxidized, resulting in the formation of acids such as lactic acid, acetic acid. Lactic acid, especially in case of muscles of human, and acetic in case of fungi and certain bacteria. And also alcohol is produced. So any of these things are produced along with carbon dioxide and energy. This energy will be less as compared to aerobic respiration. So we'll be rediscussing anaerobic and aerobic respiration. Let's first discuss what are the major steps during the respiration. So the major steps are glycolysis, intermediate reaction, Krebs cycle, electron transport chain and oxidative phosphorylation. Glycolysis is also known as EMP pathway, as it is discovered by Gustav Amden, Otto Meyerhof, Jakob Parnas. So from there, Amden, Meyerhof and Parnas, it is known as EMP pathway, and it was discovered in 1930. Here the glucose is converted to pyruvate. Glucose, one molecule of glucose, which is a 6-carbon compound, is converted into 2 molecules of pyruvate, which is a 3-carbon compound. Okay. Then it is common pathway for aerobic and anaerobic respiration. It occurs in the cytosol. Now cytosol is the matrix part of cytoplasm. If you see the intermediate reaction, where the pyruvate which is formed in the cytosol is converted into acetyl-CoA, and this reaction occurs in the mitochondrial matrix. Then next step is the Krebs cycle, which is known as tricarboxylic acid cycle or TCA cycle. And why it is known as TCA cycle? Because the first metabolite which is formed is citric acid. So that's why, and the citric acid is consists of 3 carboxylic group. So since it is tricarboxylic acid, so it is known as TCA cycle. Now during Krebs cycle, the acetyl-CoA is converted into carbon dioxide, water, NADH2, FADH2. This Krebs cycle is also known as citric acid cycle, as citric acid is regenerated by the sequence of reaction. It is discovered by Hans Adolf Krebs in 1953, for which he was awarded Nobel Prize, and he shared this prize with the Fritz Albert Lippmann, who discovered coenzyme A, which is part of intermediate reaction of the cellular respiration. And Krebs cycle, it occurs in the mitochondrial matrix. The next step is the electron transport chain. Now during this chain, the NADH2, FADH2, that is the reducing powers, they pass their electron through the sequence of electron carriers which are present in the inner membrane of mitochondria. Then the last step is oxidative phosphorylation, in which the energy of electron is utilized in phosphorylation of ADP to ATP, and it occurs in the inner you can say membrane of mitochondria. Now before we start with the glycolysis, it's important to understand these terms. Now let's understand what is ATP. ATP is the organic compound which is a nucleotide and is known as adenosine triphosphate. If you look at the structure, this is adenosine triphosphate. Three phosphates are linked here with the sixth position of the sugar, and then you can see that it is the primary energy carrier to derive various metabolic processes in living cells, such as in transportation, muscle contraction, nerve conduction, etc. Now it has got three different phosphate bond. These are high energy phosphate bond, and they are readily hydrolyzed to release the energy. So first the terminal, this one, the terminal phosphate group is removed, and then next, and so on. So when the first is removed, the ATP is converted into ADP plus PI. PI means inorganic phosphate, and then further if the bond is broken down, then ADP is converted to AMP plus PI. So on hydrolysis, these bonds are broken down. Now ATP function as energy currency for cell as it allows the cell to store energy briefly and transport it within the cell to support the endergonic reaction. ATP is produced in the mitochondria of the cell, so they are called powerhouse of the cell. ATP is produced during the cellular respiration. So ATP is also produced by the cell and is also utilized by the cell. Next is the NAD or NADP. NAD is actually nicotinamide adenine dinucleotide, which is a coenzyme. Okay, we have discussed this coenzyme in last chapter, enzymes. The coenzyme acts as a biological carrier of reducing equivalent. NAD participates in many redox reactions. Redox reactions are the one in which the reduction and oxidation occur simultaneously in the cell during glycolysis and Krebs cycle. NADP is produced by light reaction. So NADP is actually analog of NAD only, but it is produced by light reaction in photosynthesis. So it has a role in photosynthesis. NAD, when it is NAD plus and NADP plus, it indicates these are the oxidized form, and they acquire 2 electrons to get reduced. So when they have to reduce, they will acquire 2 electrons, and these 2 electrons comes from the substrate on which they are acting, plus 1 proton which comes from the substrate, and NAD plus is reduced to NADH plus H. So NADH will remain, whereas the H is liberated in the surrounding, and we will see the significance of this proton to generate the proton motive force. Now reduction and oxidation, just to revise what is oxidation and reduction. Oxidation is loss of electron, reduction is gain of electron. Oxidation is addition of oxygen, reduction is removal of oxygen. Oxidation is removal of hydrogen, and reduction is addition of hydrogen. Now coenzyme A. Coenzyme A is basically a coenzyme, mainly consists of pantothenic acid, adenosine 3-phosphate, 5-pyrophosphate, and cysteamine. It is involved in the transfer of acyl groups, notably in transacylation reactions. So wherever there is transacylation, coenzyme A plays a important role. Now we will discuss the glycolysis. Now glycolysis is a biochemical pathway in which there is anaerobic degradation of glucose to pyruvate with the release of energy, and it occurs in the cytosol of cell. Basically 3 different steps are there in the glycolysis: energy spending phase, which is called preparatory phase, splitting phase, and rearrangement phase, energy conserving phase, or payoff phase. So we'll be discussing now the reaction of glycolysis. So you can see these are the reaction of glycolysis. So come to the first reaction. We have the glucose in the cell, which is present in the cytosol, and glucose is converted into glucose 6-phosphate. So what is happening? Phosphorylation is occurring. At 6th position in the glucose, 1 phosphorus, or you can say phosphate, is added with the help of enzyme hexokinase, which utilizes, now Mg, magnesium, is the cofactor for it, and it utilizes ATP. So ATP is utilized, it produces, as energy is used up, and it generates ADP plus pi is there, which is used in the phosphorylation at 6 position. So now the glucose is converted into a glucose 6 phosphate, and this is a phosphorylation reaction. The glucose 6 phosphate is next converted into fructose 6 phosphate. Here simple isomerization takes place, as glucose and fructose are isomer of each other. So this is catalyzed by the enzyme phosphoglucoisomerase. Then fructose 6 phosphate is converted into fructose 1,6 biphosphate. So one more phosphate is added at position 1. So this is the position 1 of the fructose. Position 6 already there was addition. Now this job is done by the enzyme phosphofructokinase, which uses the cofactor magnesium, and again one more ATP is utilized, energy is utilized, and whatever pi, that is inorganic phosphate, is generated, that is used in the phosphorylation. So kinases, they do basically job of phosphorylation. Now next step, now this is called energy spending phase, because here and here the ATP are used up, okay, and phosphorylation is done. The next step is fructose 1,6 phosphate is splitted into two molecules. Now glucose, fructose, fructose 6 phosphate, these are one molecule, okay. Now when it will split here, it will split in two different molecules, okay. Fructose 1,6 biphosphate has 6 carbon, okay. So when it will split, it will split into 3 carbon molecules. So one of the 3 carbon molecule is glyceraldehyde 3 phosphate. We have already discussed the structure in biomolecules. And another 3 carbon compound is dihydroxyacetone 3 phosphate, which we have already discussed in carbohydrates, okay. So when they are splitted in two different molecules, then you can say 3 phosphoglycerate is formed, dihydroxyacetone glycerate is formed. Now what is happening over here? Dihydroxyacetone 3 phosphate is now converted to 3 phosphoglycerate. So there was already one molecule of 3 phosphoglycerate. Now 3 dihydroxyacetone is also converted into 3 phosphoglycerate. So now there are 2 molecules of glyceraldehyde 3 phosphoglycerate, okay. So this is again the isomerization, which is catalyzed by triose phosphate isomerization, and now 3 phosphoglycerate is converted into 1,3 biphosphoglycerate. So here this is the reaction where oxidation and phosphorylation are occurring simultaneously. So extra phosphate group is added, and this is catalyzed by the enzyme dehydrogenase, which is known as glyceraldehyde 3 phosphate dehydrogenase. And this enzyme uses the coenzyme NAD will remove the protons from the substrate, and it get converted into NADH plus H. Now why 2 molecules? Because here the 2 molecules of 3, we are working with the 2 molecules of 3 phosphoglycerate. So everywhere there will be double things which are being used up, okay. Now this reaction is phosphorylation and oxidation. Next step is 1,3 biphosphoglycerate is converted into 3 phosphoglycerate. So 3, what is happening over here? Here the phosphorus is removed. See, 2 phosphorus were here and one of the phosphorus is removed, that is from the position 1. Now again dephosphorylation is occurring here in presence of kinase. This kinase is called phosphoglycerate kinase, which again uses magnesium as cofactor, and now the ADP is converted into ADP as phosphorus is used up by ADP. So ADP is now forming the ATP. So 2 ATP are generated. So this is the energy conserving phase, okay. Now The 3-phosphoglycerate is converted into 2-phosphoglycerate. Simple rearrangement and isomerization is here between the molecules. And this is catalyzed by the enzyme phosphoglyceromutase. Okay, and now the 2-phosphoglycerate is converted into phosphoenol pyruvate with the dehydration. So water, 1 molecule of water is removed. Since there are 2 molecules of 2-phosphoglycerate, we will write 2 molecules of water over here. So 2 molecules of phosphoenol pyruvate are formed. Okay, and phosphoenol pyruvate is converted into pyruvate with the help of enzyme kinase. So here you can see the kinase. Again, what it is doing, it is removing the phosphorus from the substrate, and this phosphorus is used by ADP to convert it into ATP, and the enzyme is pyruvate kinase. So all the kinases, basically they are involved in the phosphorylation and dephosphorylation. Okay, so if you see the ATP calculation, so 3 phases are there: energy spending phase, splitting phase, and the energy conserving phase. So during the energy spending phase, here you can say ATP is used, here 1 ATP is used. And during energy conserving phase, you can say here 2 ATP are produced and here 2 ATP are produced. So total 4 ATP are produced and 2 ATP are used up if you see the whole process of glycolysis. So there is net gain of 2 ATP during the glycolysis. So we will say there are 2 ATP which are produced during the glycolysis. So if I write the overall reaction, the glucose is converted into pyruvate. We started with the glucose and the glucose is converted. Last is the pyruvate. So 2 molecules of pyruvate are there, 1 molecule of glucose was there. So glucose is converted into pyruvate. 2 NAD are used up during the reaction. So here you can say 2 NAD were there used up, whereas they are converted into 2 NADH plus, 2 NADH plus H plus. Okay, and then 1 inorganic phosphate which is used up here, so we'll write as a phosphoric acid, and then 2 H2O are produced by the enolase. So overall reaction, glucose plus 2 NAD plus 2 ADP plus H3 H3PO4 gives 2 pyruvate plus 2 NADH plus H, 2 ATP plus 2 H2O. So this is known as overall reaction of glycolysis. Now here you can calculate 2 ATP are produced. Later on, this NADH plus H, now this is reducing power. Okay, so it has got the 2 protons and 4 electrons. So NADH produces 6 ATP. When it will go to the electron transport chain, it is going to produce 6 ATP by passing their electrons over the electron transport system or electron transport chain in inner membrane of mitochondria. So that we will discuss or continue in the next video. Thank you.